Welcome, everyone. Thank you so much for taking time to join us this evening for a SABC educational webinar with an update to the Scleroderma Association of BC research project. My name is Kelly Grant, and I'm an SABC board member and support team leader. And I'm really very privileged to introduce our presenters, Dr. James Dunne and Dr. Kevin Keane. Dr. Dunne is a highly respected rheumatologist based in Vancouver. He is a clinical assistant professor for the University of British Columbia's Department of Medicine and specializes in rheumatology. Scleroderma and fibromyalgia are also among Dr. Dunne's clinical interests. He is a contributor to the book, The Fibromyalgia Syndrome, a clinical case definition for practitioners. He has also been published in the journal Arthritis and Rheumatology and other peer-reviewed journals. Dr. Dunne has the unique distinction of studying in 4 countries, Ireland, England, the United States, where he did a fellowship, and Canada. And Dr. Keane is a professor in the Department of Mathematics and Statistics at the University of Northern British Columbia and a visiting professor in the Department of Medicine at the University of British Columbia. He has held visiting appointments with the Netherlands Institute for Health Sciences, Erasmus University in Rotterdam, and the Faculty of Medicine at Leiden University. He has published more than 30 peer-reviewed articles in journals, 2 book chapters, and 2 editions of a textbook on statistical graphics. His research involves the development of novel statistical techniques and cutting-edge computer software for the analysis of multivariate data in the study of complex diseases. He is the co-author of articles on lung and skin involvement in scleroderma. Dr. Keane is accredited as a professional statistician by both the Statistical Society of Canada and the American Statistical Association, and is a fellow of the Royal Statistical Society. And this webinar this evening marks the kickoff to Scleroderma Awareness Month in June, when we come together to raise awareness and vital funds to support ongoing research and improve lives. And one of the important ongoing research projects we support is the SABC Research Project. I encourage you all to donate and to support our June awareness initiatives. On June the 6th, there's a group for scleroderma dance in Ladner. On June 21st, there's a scleroderma ride for research in Stanley Park. And throughout all of June, there's a Moving to Cure Scleroderma, and that's province-wide. For more details about all of these events, just go to sclerodermabc.ca. There will be time for questions. You can please stay muted during the presentation unless you're asking a question, and you're also welcome to, we'll monitor the chat to ask a question in the chat. Welcome, everyone.
Okay. Okay, let's get started. So there's our names in blue on white. And the next thing I'm going to go to for the next slide is just run through the agenda. So Jim and I are going to give you an introduction, and then we're going to move on to discussing research goals, what the principal investigators have been up to in terms of their research projects, what the graduate students have been doing with their research projects. We're going to discuss ongoing research, stuff that hasn't been published or presented yet, and we'll basically wrap things up with a lay summary with an opportunity for questions after the lay summary, but you're more than welcome to ask questions during the presentation. Kelly's going to be monitoring the chat box for us, and she just has to bring something to our attention, and we will stop what we're doing and address the question. So here we go. By way of introduction, the SABC research program began as a single project, and then it transformed into a series of projects, and then it morphed into multiple parallel projects that we call research modules. For those of you who aren't aware, the program is responsible for managing the Scleroderma Biobank, which is at St. Paul's Hospital in Vancouver, and that began as a limited shelf space in a -80 degrees Celsius freezer. The Scleroderma Data Bank is hosted at the University of British Columbia on the Vancouver campus, and it began as a single spreadsheet in an encrypted Microsoft Excel file. So the program has grown quite a bit and quite much from its humble beginnings. We have over 600 participants in both the Scleroderma Biobank and the Scleroderma Data Bank. The SABC Research Program works with researchers at the University of British Columbia in Vancouver, at the University of Alberta in Edmonton. In a week's time, I'll be heading to Edmonton to work with one of our colleagues there. We're active with SPIN, the Scleroderma Patient-Centered Intervention Network, and we're also active with the Canadian Scleroderma Research Group, which is now based out of Calgary under Maggie Larche. Program members are active in presenting their work for peer review through presentations at international and national biomedical conferences. There's a national conference coming up in Calgary at the end of June, and also through submission to international biomedical journals for publication. Research goals. Stated simply, the primary research goal for the Scleroderma Association of BC Research Program is to find a functional cure for scleroderma. Now, a cure for scleroderma is if we found one pill that you only had to take once in your life and you were cured of scleroderma. A functional cure is something where you might have to take a pill or two every day for the rest of your life and that all the symptoms of scleroderma disappear. So the enabling research goals to obtaining our primary research goal is finding and validating biomarkers in peripheral samples to track and predict disease progression in people with scleroderma with respect to things such as interstitial lung disease, calcinosis, and food malabsorption. Principal investigators, well, we've already had Jim's introduction. Jim's in the UBC Division of Rheumatology, and so is Hayne Kim. In the UBC Division of Respirology is Dr. Christopher Ryerson and myself. And from the UBC Department of Anesthesiology, Pharmacology, and Therapeutics, we have Dr. Tilly Louise Hackett, and we're presenting some of the work that done by students that Tilly and Jim are jointly supervising. And our second partner in the Department of Anesthesiology, Pharmacology, and Therapeutics is Dr. Amrit Singh, biometrician and bioinformaticist. So let's start with principal investigators research. So the first thing we're going to look out of two items is looking at a presentation that was made at the World Scleroderma Research Conference this past March in Athens on distinct metabolic profiles that differentiate scleroderma with ILD from idiopathic pulmonary fibrosis. This was done using blood samples from voluntarily and written consented patients and controls, among them 63 SSC patients, of which 17 had limited cutaneous scleroderma without ILD, 18 had limited cutaneous scleroderma with ILD, 10 had diffuse cutaneous scleroderma without ILD, and 18 had diffuse cutaneous scleroderma with ILD. In addition to the 63 SSC patients, there were 16 IPF patients and 13 healthy controls. And this data was analyzed by chemical isotope labeling, liquid chromatography, mass spectrometry, simply known as LCMS. We said we wanted to differentiate the IPF patients from the scleroderma patients. And so we have an interesting graphic here, and I'll let you observe it for a couple of minutes. We're looking at two artificially generated axes from a factor analysis done on individuals for all the metabolomic data. We have color coding here. Controls are in green, and you'll notice that all of the controls are separated from the IPF patients and most of the scleroderma patients. If you go to the red, that's diffuse scleroderma patients who have interstitial lung disease. They're in the upper left-hand corner. And just below them, in orange, are the diffuse scleroderma patients who don't have ILD. And then further down from them in dark blue are the limited cutaneous uh, scleroderma patients with ILD, and closer to the controls with the weakest of the diseases are the patients with limited cutaneous scleroderma and no ILD. And if you look in the upper right-hand corner, completely separated from both the controls and the SSC patients are the patients with idiopathic pulmonary fibrosis, or what we call IPF. So we were able to see a clear separation. But that wasn't the only thing we were looking for. We had a major finding, and that was we found that the following were upregulated in patients with diffuse scleroderma and interstitial lung disease compared to controls in patients with IPF. We found that the arginine and proline metabolism associated with cis-4-hydroxy-D-proline, or CHDP for short, um, these pathways were upregulated in patients with diffuse cutaneous scleroderma and interstitial lung disease, suggesting autoimmune skewering of lung tissue is more of a feature of ILD in patients with scleroderma than aging and lung epithelial lung cell injury in IPF patients. So we speculate that CHDP could identify patients with scleroderma who are at a high risk to develop ILD before it even shows up on imaging, and those with ILD who could become fast progressors.
Can you go back and let me talk?
Sure, go ahead, Jim.
So go back to the
The
the data
graphic.
slide. Yeah.
There we go.
So okay. So basically, you're looking at yourselves because many of you are part of this study, and you will appear here as a number, but I don't know what your number actually is. So that's the first thing. The second thing is what we were looking for is a robust biomarker, that is a symbol that would tell us what the disease was, where it was in terms of its progression, and it would differentiate itself from the other subtypes. And who cares if that's the case or not? Well, the further upstream you can go in terms of diagnosis, in other words, the earlier you can make a diagnosis and the earlier you can make an intervention, a therapeutic intervention, then the more likely it is that you will be able to prevent ongoing damage or irreversible damage. So what came out, three interesting things came out of this. The first one was that, as you can see, they differentiate pretty well, especially normals and idiopathic pulmonary fibrosis from interstitial lung disease with scleroderma with interstitial lung disease. So that's really interesting. And the other ones are clustered around the middle, and they're not sure who they are, but that's not such a bad idea because many of these are either without interstitial lung disease or they're limited. So that was the good news about that. The second thing it showed was some of the metabolic. So the other thing we wanted to look at pathophysiology, and it showed some of the metabolic pathways that were abnormal that I won't go into, but the arginine proline one was significantly abnormal, and that if you can reverse that, that would be an interesting thing to be able to do. And the third was the most. If you go forward a slide,
Here we go.
thanks. If you look at cis-4-hydroxyproline, which is a, a breakdown product of collagen, if you look at that, then it occurs early in the disease and is a very robust marker. But what's also interesting about it is that it is also metabolized, or collagen is metabolized in the gut and may produce cis-4-hydroxyproline, which means that one of the things that we're interested in looking at now is what contribution the microbiome in the gut makes towards this metabolic pattern. And if you can reverse that, that would be also quite interesting. So that's a quick brief a reason or overview of what this shows. Where we're going next with this is we're going to look at another cohort, a confirmatory cohort is called. And the second thing we're going to look at is collagen turnover to see if we can see when collagen turnover begins and if this will mark it for us. And the third is to look to the relationship with the microbiome. So those are going to be really interesting projects if we come to fruition with them. Okay, sorry, go ahead.
Thanks. And collagen is going to be featured on a couple of slides later on in this presentation. Okay, so we are up to date with that information. Thank you, Jim, for filling in those details. And here we are. Here's the other project that they've been working that Jim and I have been working on. And this material was presented at the European Alliance of Associations for Rheumatology in Vienna back in June of 2024. And where we are with respect to this research right now, as of today, is I'm wrapping up the manuscript that is in preparation for review by all the other authors and then for submission to a journal in Europe. So what we were looking at in this research, and if you're interested in following it up and reading it, you can see there's an HTTP link there. And hopefully these links will be made available on the website, which will also be making the recording of this presentation today available, that you'll be able to find those there and have a quick connection by clicking on them. So this work concerned DNA methylation estimates of hyperaging and systemic sclerosis in idiopathic pulmonary fibrosis, or to put it simply, scleroderma and IPF are both diseases of advanced aging, that people are older physically than they should be according to their chronological age. And for this study, we had, among the people watching here today and participating and listening to us for this presentation, 51 scleroderma patients, slightly different group than from the previous slide, slightly different numbers, but closely the same. 13 IPF patients and 12 healthy controls. And what we do in our studies, which is a little bit unique, is we have two control groups. Well, healthy controls, that's one control group, and we use IPF patients as another control group. So we're bracketing scleroderma patients in between the healthy controls and the IPF patients, which tend to be worse off quicker in the course of their disease. So peripheral blood samples were analyzed using the methylation epic B chip and assessed for the accelerated pace of aging using the Dunedin pace metric. So Dunedin pace. Dunedin is a town in New Zealand where researchers have basically worked very hard to catalog and map this metric to participants in their study from New Zealand. And pace refers to the fact that compared to other measures of biological aging, this measures the change, the speed of aging, not just the actual age. That's done by other measures, the second generation measure. This is a third generation metric, and what it measures is the speed of aging, which can change over time. Okay, I just had a hang up on my mouse. Don't worry about it. Okay, so we fitted a multiple linear model to the data. So we have created an estimator of Dunedin pace from the available data using Dunedin pace to create the model. And we were able to come up with these results. So again, we've got our color coding for the 6 different categories of patients and controls. And we also have, in addition to the color-coded actual observations there of actual age and the fitted Dunedin pace, we also have simple linear regression, simple lines going through the plots of data for each of these specific categories in this figure. So for example, if you look to the bottom, you'll see a blue line segment. That's kind of like the best linear fit between all of the control data for the pairs of data for fitted Dunedin pace and age. Now, optimistically, and we're doing better than optimistically, healthy people should have a Dunedin pace measure of 1, meaning for every year of chronological age, they have 1 year of biological age. But we're dealing with people from Vancouver who are probably fitter and eating a healthier diet than the people who were in the original Dunedin study from New Zealand. And we note that virtually all of the data points collected for the controls are beneath 1.0. But we do see a trend as people age from 20 to 70, the Dunedin pace increased, linear relationship. But if you look at it carefully, it's still below 1. In fact, if you look at the limited scleroderma patients in this study, you'll see that the inverted green isosceles triangles, only a few are above 1.0, And the best fitted line, although it increases with age, a slightly faster rate and at a higher level than the controls, is still below 1.0. If you go to the red color, these again are scleroderma patients without interstitial lung disease, but these are the diffuse cases. So they're aging more rapidly. And the rate of change of the pace is also slightly more rapid. If we go to the gray diamonds and the gray line segment, this is for the limited cutaneous scleroderma patients with ILD. So these patients, of course, we can see the gray line is worse off in all assessments compared to the green line. And we see that the aging is greater than 1, and it also increases as the patients age. And the next color that we see that's represented by a line segment just above them is for the patients with idiopathic pulmonary fibrosis. They're also starting out with a higher mean level of Dunne pace and with a greater rate of aging, because the rate of aging is increasing as time goes by. And then finally, the patients with the highest level of Dunne pace are patients who have diffuse cutaneous scleroderma and interstitial lung disease as well. But if you look at how steep the lines are, and which the steepest ones, it's clear that the IPF patients are, as we would say, decompensating more rapidly. They start out, except for the diffuse cutaneous scleroderma patients with ILD, at the higher levels, and they more rapidly age, for lack of a better way of describing it. In addition to that observation that we made, in the statistical model of the data, we found that being male increased Dunne pace by approximately 5%. Having stopped smoking will decrease Dunne pace by about 20%. And never having smoked decreased Dunne pace by nearly 25%. So these were the most important findings that we found with respect to the basic demographic variables. Jim?
Sorry. So first of all, there's good news and bad news. The good news is we're all aging, and the aging clocks are ticking away. And some of them the biological clocks are more aged than our time from birth. There's in IPF, there's been an interesting insight into that, is because they looked at the rapid aging in IPF, and then they went and looked at senescent cells, which are also known as zombie cells, and discovered that there are more senescent cells in these patients in general, but also in their lungs. And subsequently, a group, I forget where they're from, I think in Europe, went and tried to remove the senescent cells from the IPF patients' lungs by killing them off. And they did just that, and they slowed down the rate of progression of fibrosis in the IPF lungs. So it looks like it was a good treatment for the IPF lungs. The second thing is that although your biological age may be different from your chronological age, it may be possible to revert those back to the normal rate of aging. And lots of studies have showed that if you have a biological age, it's not written in stone. It's mobile and can either go forward or go backwards, relatively speaking, in time. So it is possible to change the biological age. That is the degree of methylation of the DNA.
I have a
So there you go.
question from the chat.
Go ahead.
The question is, were Dunedin pace IPF patients older than the scleroderma and control patients?
No, that was a population-based research project, and like us, they covered a broad range. But if you look at the plot here, you can see, for example, that our IPF patients are older because there's a later onset for that disease, especially compared to scleroderma patients. So, typically for IPF patients, onset is after age 50 in men, and typically in women, the onset for scleroderma is around 50, and more women have scleroderma than men.
Okay, thank you.
And men are at a disadvantage by having only one X chromosome, and men age faster. And that's what we found here, that the pace of aging for a male is 5% more than a female. Now, we weren't just looking at the basic demographic variables. We had plugged in other information, such as, for instance, a particular cytokine known as interleukin 2, which is strongly associated with inflammation, which isn't necessarily a bad thing if it leads to an uncorrupted repair mechanism. But if it persists in being elevated too long, then you're going to have a situation where the repair mechanism in the body, the skin in the lung, is affected by higher levels of inflammation that's really needed. And we refer to this as inflammaging, which is due to inflammation causing increased aging. So we found a very small but noticeable association with that. So that's on that side. The other thing that we put into the statistical model was everything we knew about what the patients were taking with respect to drugs to control their disease. And again, you're not just taking one pill and it's not reversing all the effects. But we did find that receiving nintedanib reduced Dunedin pace by nearly 13%, which is rather significant. And nintedanib was first introduced for IPF patients to at least slow injury in the lung. And then based on that experience, clinical trials were conducted in SSC patients with ILD, and it was found to be as successful. The jury's still out on how successful nintedanib is in controlling and reversing damage in the skin of scleroderma patients. So when we're talking about treating people, think of that picture that's back there. If we're going to have a functional cure for scleroderma, it's going to have to change that aging curve and reduce the overall values of the Dunedin pace. So that goes hand in hand with a functional cure. And you're going to see that in coming slides, interleukin 6 comes up again. Okay, so enough for the principal investigators. Let's turn our attention to what the graduate students have been doing. And here are two of our graduate students. They're both PhD students in the Pharmacology and Therapeutics program at the University of British Columbia. They're both co-supervised by Tilly and Jim. And they are working on quite closely related projects. They're all, both of them, Brandon and Ravine, are working with fibroblasts, but with respect to different axes. So Braden's focus is on looking about the situation involving platelet-derived growth factor signaling. We're going to call that PDGF for short. And Ravine's work is focused on the role of transforming growth factor beta and its inhibition as a potential treatment to reduce fibrosis or scarring in scleroderma. So her work investigates the role of each of three types of TGF-beta in activating fibroblasts from skin biopsies and lung tissue samples to determine if inhibitors of TGF-beta will reduce collagen production by fibroblasts. Both of them are interested in therapeutic strategies that can slow disease progression and alleviate patient symptoms. Okay. I have a little hang up on moving to the next slide. There we go. So this is where we are now in terms of the treatment of scleroderma. It's called the pan-inhibition approach. We have a hammer and we smack it down and we hope that it's going to work on everything. And one very good example of a pan inhibitor is nintedanib, for slowing lung deterioration in interstitial lung disease in scleroderma patients and lung disease fibrosis as well in patients with idiopathic pulmonary fibrosis. So it's trying to do many things at once, and that has some consequences. One of the consequences of that is when you're trying to treat all tissues and all isoforms identically, that leads to high side effects and limits dosing efficacy. Okay? So it's not going to work that well, and it's also going to cause side effects in the patients being treated by it. The efficacy graphs show that broad inhibitors show some effect in the lung, but as I said, the jury is still out on whether we're seeing an nintedanib resolved skin fibrosis in scleroderma patients. And here's the kicker. Fibroblasts are not uniform, so to stop progression of disease in scleroderma and IPF patients, we must target distinct biological variables. That is the tissue, the pathway, and as you'll see in a slide or two, the isoforms. And we've had 2 recent publications as of last month. We have the links to those papers if you're interested here, and hopefully they will be available on the SABC website to the 2 papers. Brandon's research is in the first paper here, as he is the first author, looking into PDGF factors on neonatal-derived dermal and fetal-derived lung fibroblasts. And the second paper with Ravine as the lead author is her PhD research, where she's looking at TGF-beta isoforms in normal dermal and lung fibroblasts and made some important identifications as which of those fibroblasts seem to be more important than the one that's classically been studied the most. We thought it would be worthwhile to show you what the students are actually doing. And we asked the students to put together these couple of slides. I'd like to bring your attention to the images for the column denoted dermal fibroblasts and the second column alongside to the right called lung fibroblasts. So this is what they're seeing through the microscope. Where it says A, no cells, that's the medium, and that's the microscopic picture of what's on that little spot on the slide. Okay, and then what happens to that medium in these 6 examples in the first column after that is fibroblasts are introduced and cultured. And different things are done to them. So if we look at control, those fibroblasts have been placed into the culture and grown. And you'll see a cloud in there with an arrow pointing to it. So that cloud is 3-dimensional collagen gel. Under conditions of no stimulation with PDGF isoforms, that's how much growth you're going to get in contraction you're going to get in the gel of 3-dimensional collagen. Next to it, it says PDGF AA. Well, AA is an isoform of PDGF, and as you can see, we've got a nice blob there, but it's showing more contraction than the control. Next one over, PDGF AB. More contraction than control, possibly more contraction than PDGF AA. And in the bottom row, we have PDGF BB and PDGF CC, and finally PDGF DD. And all that information is summarized. That's just one set of experiments, is summarized in the bar chart below, where the collagen 1 contraction is measured. And we've got the asterisk there indicating that we're seeing statistically significant. And if you look at the change in the levels there, those are practically significant differences between the treated dermal fibroblasts and the untreated control fibroblasts. So we're seeing greater contractions due to all of the different isoforms for PDGF, but more strongly for every one of those isoforms, expect PDGF AA. Going over to the next column, we have the same setup, only this time we're growing lung fibroblasts and observing the contractions. And below, you can see the collagen 1 contractions for all the samples that have been done by Brandon. And as you can see, there are statistically significant differences as well. And generally speaking, in healthy controls, in lung fibroblasts obtained from healthy controls, that we're seeing greater collagen 1 contraction once any one of the isoforms is added from PDGF. But as you can see, there are differences among the levels of response according to isoform. So that's a solid contribution to basic science from Brandon. Brandon also measured interleukin 3, pardon me, interleukin 11, which is another cytokine, and like interleukin 6, it's involved in inflammation. And as we can see from slides A and B, if we treat fibroblasts with PDGF isoforms and measure the interleukin 6 that we're getting in healthy fibroblasts, an increase in the amount of interleukin 11 produced, and from these in vitro lab experiments on plates, we extrapolate to patients and saying, okay, if we're seeing increases of PDGF in patients for any of the isoforms, except perhaps for PDGF AA, that we're seeing an increase in the concentration of circulating interleukin 11. The other thing we looked at was the amount of expression of collagen 1. And now we're switching over to Ravine's work involving the 3 isoforms of TGF beta, 1, 2, and 3. As I said, 1 has been the most studied of the 3. Next, 2, and TGF beta 3 has hardly ever been studied. So this is an important contribution by Ravine because she's providing information about TGF beta 3, which is what we don't have in the literature. Beta 1 and beta 2 have been quite carefully studied. So again, we're seeing separately for dermal fibroblasts on the left and lung fibroblasts on the right that have been cultured on slides, we're seeing the amount of normalized collagen 1 protein expression. And if you take a normal fibroblast and add beta 1 to it, you have an effect. Beta 2, you have an effect. And beta 3, you have an effect in the dermal cell line. If you go to the lung fibroblast line, you can see that there is a very significant difference between the various isoforms, where the normalized collagen protein expression for collagen 1 is higher for beta 2 compared to beta 1 and beta 3 compared to beta 2. So the isoforms are not identical. And so if we're going to be working at reducing overproduction of collagen, we have to bear in mind that there are 3 different isoforms that somehow must be controlled. Now, Ravine also looked at the amount of circulating interleukin 11 produced by the dermal fibroblasts and the lung fibroblasts. And we found that if you take a control fibroblast and add any one of beta 1, beta 2, or beta 3, that's going to cause an increase in interleukin 11 expression. That's in the top row, in the lung on the right and dermal on the left. And if we switch from interleukin 11 to interleukin 6, which we've already discussed, we can see that there are positive effects in healthy fibroblasts. If you add beta 1, beta 2, and beta 3, it looks like we're upregulating both the interleukin 6 and interleukin 11 inflammatory channels. So if we take all the results from the 2 students and put them together, what are we finding in the so-called isoform pathogenicity matrix? What are the bad actors? And the bad actors are in the darkest colors. So the ones that have the most effect on collagen production and on interleukin 6 and interleukin 11 production and on contraction are TGF beta 2 and 3. And from the 5 isoforms for PDGF, it's the double B. So let's recap that. So we have a unified dual pathway theory of fibrosis. On the one side, we have a gear powered by PGF double B. That's the inflammatory contractile engine. That's on the left. And on the right cog, we have the structural engine that's being powered by TGF beta 2 and beta 3. So that's the major findings that we're taking out of these two studies. Put together, that's what we're seeing.
So let me comment on that, if I may. If you
Go
go
ahead,
ahead,
Jim.
Garrett. Yeah. So one of the ways of looking at scleroderma, if you forget for the moment that's an inflammatory or an autoimmune disease, and think of it as wound healing gone wrong or gone awry. And this is where wound healing continues and doesn't stop, and that's where the fibrosis and the scarring comes. And TGF beta is produced by fibroblasts and is the main driver, or not produced by fibroblasts, it is actually, but produced by other cells which turn on fibroblasts to produce collagen, and the collagen is laid down to make the scar. The PDGF is produced earlier on in the cycle, which is activating cells and make them proliferate. And then you got more fibroblasts and more other cells proliferating, and then they make more collagen, and you make more scar, and you're eventually they die off, and the scar stops, and that's how you have normal scarring. But that's not what happens in scleroderma. And the other thing that's interesting from here is that most of the medications that have been used and are being used are not looking at blocking TGF beta 2 and 3. And those slides that you saw, which you may not have noticed, are much more powerful at turning on fibroblasts to produce collagen than TGF beta 1. So they need to be addressed. So that's one of the interesting things that we found. And the other interesting thing that we found was that the PDGF for cell proliferation, it wasn't just PDGF, which the medications currently like nintedanib block, but it is a subset of TGF, which is the BB, that needs to be blocked specifically. So what happens here is that we now see a more granular change in the process of developing inflammation and scarring, and a more focused look at the products that are driving that. And that leads to a whole different way of looking at how we would stop them. Now, how they're initially turned on is a whole different question. And if anybody asks me, I'll be happy to answer that, but we'll move along because we're nearly 40 minutes in. Go ahead.
Yeah, so just to quickly recap, if you shut down the PDGF axis, the tissue stops contracting but continues to build massive structural scars through collagen 1. If you only inhibit TGF beta, the scarring stops, but the tissue remains heavily inflamed, contractive, and hyperproliferative.
So that's the good news from our study, and that's pretty much from our study. Okay, go ahead.
Okay, we got a little glitch again in terms of, there we go, moving things back. Oh, too quick. There we go. There. So let's talk about ongoing research now. For Doctors Dunne and Keene, stored on the SABC, I'm on the right slide here, hold on. It's getting a little, moving slides with the mouse is a little bit problematic. And that's what the new system is doing with Zoom. Okay. There we go. So let's talk about ongoing research. This is the work being done for which there are no published abstracts or manuscripts for the postgraduate students and Dr. Hackett. And the principal investigators are working to acquire tissue samples of removed diseased lungs to grow fibroblast cells in collaboration with transplant teams at Vancouver General Hospital and the University of Alberta Hospital in Edmonton. This research needs to be broadened from fibroblasts cultured from the skin of healthy controls and the lungs of healthy controls to include fibroblasts from patients to assess treatment benefits in vitro in the lab of the single molecule drug galunisertib, which was designed initially to inhibit the TGF-beta receptor 1 kinase and so targets TGF-beta 1 signaling, which we showed on the preceding slides, to reduce fibrotic scarring of lungs and skin. So these are the first images, breaking results from Ravine and Brandon. These are showing fibroblasts grown from the skin of 2 study participants with both with diffuse scleroderma and interstitial lung disease. These are microscope pictures through the microscope. The different colors are there because different light sources were used for the 2 samples. It doesn't mean that there's any specific difference between the 2 samples whatsoever. So this is brand new results. Their 2 publications dealt with healthy controls, and we're starting to move forward with their next phase of analysis, where we're going to see how these factors actually behave in fibroblasts grown from skin and lungs of patients who have either scleroderma or IPF. For Doctors Dunne, as far as ongoing research, stored in the SABC database are microRNA sequences, other small and long RNA sequences, and DNA sequences. And from this, we're going to be developing an abstract and a manuscript on small and long RNA sequences in scleroderma patients and IPF patients. We're going to develop an abstract and manuscript on DNA sequences, merge the data from the 3 genomic sequences to provide justification for using microRNA sequences as therapy to add beneficial microRNAs and to reduce harmful microRNAs, and possibly in addition to that, not just as therapy, but as biomarkers. As stated, the primary research goal of the SABC research program is to find a functional cure for scleroderma. So now we're switching gears from a more technical presentation to a lay summary. So let's get into that. Several specific outcomes of a number of research models have just been presented after peer review leading to publication of abstracts and articles. But I think it's hard to see how all these can tie together in a cohesive way. Other than ongoing research projects that have been discussed, it's hard to see how this relates to the published abstracts and articles. So everything needs to be tied together as to how all the work we're doing with these different research modules relate to the primary research goal of finding a functional cure for scleroderma. And we're going to attempt to do this in the next couple of slides in a comprehensive synthesis. Okay. Right. So let's talk about synthesis in a nutshell. Scleroderma, interstitial lung disease, and idiopathic pulmonary fibrosis are molecular diseases that need to be assessed by molecules before being treated with molecules. The designed experiments and collected data of the SABC research program on molecules are thought to be associated with inflammation in the body, thought to be associated with premature aging of the body, thought to be associated with fibrosis scarring and faulty tissue repair in the body, and associated with the immune system attacking the body, something we call autoimmunity. The SABC research program is holistically and ambitiously looking at all possible molecules involved in damage or involved in repair, naturally occurring in the body or not naturally occurring in the body, but could be introduced into the body as therapy. The SABC research program is a large task with an approach that examined molecules known to be associated with or reasonably expected to be discovered to be associated with the inflammaging fibroautoimmunosome. Have a very long word encapsulating what we're trying to do. And what are we trying to do is collect flaws that result in tissue damage instead of tissue repair and regeneration. And thank you very much for your time, and I think we're open for questions.
There are 4 questions in the chat. Do we have chat?
No, but I have a question that was sent to me, somebody who couldn't attend, and I don't know if you can answer it or not because it's not really the focus of what you said tonight. But the question was, will taking a low dose of sildenafil orally be of benefit to people with scleroderma and Raynaud's?
Certainly the Raynaud's and tadalafil is better than sildenafil. Sildenafil is cheaper, but it doesn't last as long. Tadalafil is a little more expensive and lasts for 24 to 72 hours. They both are micro vasodilators and they block PDE5. So yes, they're very powerful in helping with Raynaud's phenomenon and keeping the blood vessels trending towards being more healthy. Yeah.
Thank you.
I have a question. Kevin, you had mentioned in either one of the slides or when you were just explaining that the onset for women is 50 years old. Did
No,
I get that
30,
right?
approximately 30.
Okay. So
Yeah.
we get a lot of phone calls where the people are being diagnosed a lot earlier than 30. Is that a number that's just stayed for a while or, and has it been looked at?
I don't think anyone's done any studies on whether we're seeing women present at earlier ages or we're just doing a better job of detecting it earlier, and it was there before, but we were picking it up six years down the line instead of at 23, say at the age of 29.
Okay, makes sense. Thanks.
So the biggest data sets are the USTAR data sets in Europe. We're in the thousands, and they update them every year, and then they report on their updates. So they're talking about the median number, I guess. So it hasn't changed that much, at least not in Europe. But as you know, the disease varies with different populations. The Japanese are somewhat different to the Europeans, and the Americans are different, or the North Americans are different again. So it's a depends on where you're at, I guess.
I have a couple of questions in the chat. One is,
Yeah.
based on these findings, how long are we looking at for an actual treatment?
Well, first of all, you have an actual treatment, but it's an intermittent treatment. How long are you looking for a better treatment? So the short answer is probably not that long. We're just talking about our work here, but there's an awful lot of people doing an awful lot of work. The most exciting work has been the CAR T cells, and there are 3 phase 3 trials have just started, and they are showing very interesting changes in scleroderma where they can stop the fibrosis in its tracks, it would appear, but they have to confirm that. And if that's the case, our job is then to see how you can reverse the tissue back to normal. So that's the first thing. Secondly, there are a slew of new drugs coming in. One of the first drugs for TGF beta 3 is also in a phase 3 trial. So that's come up. And we're getting better. The field is getting better and better at getting more focused medications that work better. So, for instance, nintedanib is a first-generation antifibrotic, which, if any of you have taken it, will know it has got awful side effects. But the next generations out will be better than that. So, I don't know that you'll find a single magic pill, although CAR T may be it, but it'll be like rheumatoid arthritis or HIV, where it turns into a chronic disease that will need chronic maintenance or other therapy and not one that you can simply walk away from. I hope that answers your question. So the answer, 5 years.
And that's similar to the next question, which is how far away are we from the functional cure that's being researched? So I think that's a similar answer to that.
Well, we're longer than 5 years, but then
Longer
send
than
more
5
money
years,
into it.
yeah.
Yeah. So, and also the field is changing so rapidly in the way the amount of data we can analyze and how quickly we can analyze it, and the amount of experiments we can do, and of course the dreaded AI is all over that in terms of making a difference to that. So that makes it in terms of drugs now going from benchtop to phase 2 trials, which used to be 5 to 7 years. The most recent one was out of Oxford for IPF, and it went from concept to phase 2 in 18 months.
Wow.
Phase 2 is where you take patients and look for therapeutic efficacy before going on to the major study.
And to give you a backgrounder of how fast things are changing, so we presented our methylation and hyperaging research 2 years ago this June in Europe. We're wrapping up the paper now 2 years later, and I've been using artificial intelligence to plumb the relationship between hyperaging and 24 autoantibodies and 99 cytokines simultaneously. And so that's leaps and bounds ahead of what we actually presented 2 years ago. So the pace of change just on the artificial intelligence side is amazing, and the speed is amazing as well.
Thank you. Another question is, what's the best way someone with scleroderma can support the research?
I would say
So
participate in research, read the consent agreement, and if you are capable of voluntarily consenting, provide tissue samples. And Jim, I think we're
and
looking
money.
for skin samples, are we not, in the near future?
We're still looking for skin, but certainly become associated with SABC because they do outreach and are a major support group and educational group for people with scleroderma and a portal into the system. So if you can help out with them, that's the first thing I would do. The second is if you are interested in a particular project or some of the projects that we're doing, if we need samples like blood or whatever, then and more recently stools, then talk to us and we'll be around to your door in about 15 minutes. And then finally, like coming up next month, if you can help SABC generate funding for these programs, that would also be very helpful.
Thank you. That's excellent. A question here is, and I agree with this question. I have this question also. Is there any hope for calcinosis?
Yes, but not easy. So calcinosis is difficult, and we've just started to get a handle on it. And I think if it's an upstream, if we can get upstream of the calcinosis and prevent it happening at all, that's the best outcome, and that's what people are looking at. So that things like tadalafil and sildenafil, which improve blood flow, if that's used earlier than it is currently, may protect you from getting calcinosis because it protects you from the ischemia. And when you get ischemia, then the blood vessel gets damaged. And when the blood gets damaged, your body tries to heal it. And if it's a chronic healing, it'll cap it off with a little bit of calcium to make sure it doesn't break down again. So that's how that works. And there are ways now, there are drugs that can reverse some of that calcinosis. And they've been showing that in atherosclerosis, but not in scleroderma as yet.
Okay, well, that's hopeful. Thank you. Another question is, where can we get more information about the CAR T cell research?
It's everywhere. So the CAR T research, you can look it up. I would think the US Scleroderma Association, whatever it's called, would have it on its website for sure. Maybe the Canadian one does as well. The most recent scleroderma, ninth scleroderma Congress at Athens had many presentations on CAR T cells. So their book of, if you can look up their book on abstracts and presentations, you will get it. And the easiest thing to do is to go ask Google. Don't ask Gemini or Claude or whoever to please update me on CAR T cells, and you'll get a nice long, and with citations, make sure you tell me you want citations, otherwise they'll hallucinate, and they will give you a summary.
So the link we have for our presentation in Athens is not just to our abstract, it's to all the abstracts presented. And you will find the abstracts there that were peer-reviewed and accepted on CAR T cell research. My impression from being at the World Scleroderma Conference in Athens in March was we're kind of like going beyond research, and CAR T cell therapy is becoming an accepted therapy. So the next question is, okay, which centers in Canada are going to be offering this?
Yeah, for sure. That's the next question.
Yeah.
Another question here is, are there opportunities to participate in research in Edmonton? This is one from one of our.
Yes, there are. I've spent about a third of my time during my sabbatical this year in Edmonton. We have a close colleague there, Dr. Mohammed Osman, who's a young researcher. He's getting his research program established. He's at the University of Alberta Hospital in Edmonton, and he is uniquely placed both in two departments, or divisions, I should say, Division of Rheumatology and the Division of Immunology. And he is actively recruiting patients for a trial in scleroderma patients at this point in time. And we're looking to collaborating with him here in British Columbia and having our patients be involved in that study as well.
Okay, thank
So
you.
stay tuned from the SABC webpage, and we'll update you on our side and make sure that there's a link that could be available for people of interest in Edmonton and Alberta that they can contact Dr. Osman's research team directly.
Excellent. Thank you. Any other questions? There's
I
nothing
have,
else.
sorry, Kelly, I have another
Yes,
one.
okay.
At our conference, you were looking at and wanting stool samples, and then we sort of followed up, and then nothing ever happened. Are you back on wanting the stool samples and
Yes.
do the,
So we've started to ask people that go to the combined clinic, so it hasn't actually totally gone away. I was on two minds as to what to do next, whether to blitz you all and like get 60 people all at once, but I'm not quite sure whether I should do that or not. But yes,
so
we're still
reach out
interested.
to, you can reach out to me or reach out to Cecile,
Yes.
and we'll try to get some bodies down to you or whatever it needs to be done.
So maybe when I'm down to, at the walk and cycle for scleroderma, and I sit and eat for scleroderma, maybe we can talk about that and maybe get a
Okay.
project going for the fall, because I would really like to do that. That if you
Okay.
can help out that way. Okay.
Great.
Sure.
Thank you. That's great. The Susan says that we'll be in touch with Dr. Osman. Thank you for a robust presentation. And the next question is, has the research revealed the causes?
I'll take that one. The short answer is scleroderma is not a hereditary disease. It's not inherited. We know that from twin studies. It's a genetic disease, but then all diseases are genetic because they all involve molecules produced in our body by our genetic assembly. But there are other molecules that are entered in from the environment. So one of the studies that we were pulled into originally and then had to seek other confirmatory pathways was involving microRNA. So microRNA is not something that you inherit from somebody. It's in the environment. You can get it from a mother, you can get it from a father, you can get it from a brother or a sister. And it's not well understood the full impact of microRNA on cause of disease and disease progression. But we've got an inkling of what's going on, and the work that we're going to be doing with the long and short RNAs and the germline DNA all fits into the puzzle of trying to determine what the underlying environmental cause might be for scleroderma.
So,
Okay.
there are lots of candidates, but no smoking gun, I think is the summary. And I think it's fairly obvious now that we know that for most people, the innate immune system is triggered. That's the tripwire that our bodies use to as its first line of defense against organisms, against chemicals, against whatever it sees as a threat. And it's a fairly non-specific, but it's well known that it's triggered in scleroderma, and it releases IL-1, which is a strong trigger for TGF-beta. But what they are, there's CMV possibly may be involved, some chemicals may be involved, silica dust may be involved. All of those have been looked at, but none of them have been just the only involvement.
Okay. Okay.
And just to follow on that a bit, a little bit for the presentations that I've done today with all the research that I've shown from the various research models. It shows that we are looking at the pathways, the molecular pathways that are involved in disease progression and maintenance in scleroderma and IPF. So while research has not really revealed the causes yet, we're getting closer. We're getting a much better picture now than we had 20 years ago about what pathways are involved. So you saw that Ravine and Brandon were doing their work on the PDGF and the TGF beta pathways, and Jim, he's got the lead paper on the metabolomic pathways. We're talking about environment. Well, we get food from the environment and it goes into our bodies, and then our bodies react with that food and creates any one of a number of hundreds of thousands of metabolomic products. Which ones are implicated in disease? And we threw out a couple right there. Taurine, hypotaurine, and arginine, and proline came up. And then there's the work that's being done on methylation, which is another series of channels having to do with advanced aging as a result of taking our human chromosomes as we age, and the ends of the chromosomes tend to be more tightly wrapped, and we're producing less of the good chemicals that we need in our body, or not at all, which is responsible for what we observe as aging and a sensitivity to opportunistic diseases. So we're doing very good on pathways. We're getting closer on what the cause is.
Well, that's hopeful. Thank you. I don't know if there's any more questions, but we're approaching 8:10. Does anyone else have any more questions? Okay. I'm going to stop the recording. Here we go. There we go.